always done about is always done with respect to a concurrent control group for that particular agent. So it sticks to that basic principle. There's actually a lot of interest in the context of these trials of trying to use historic information. So, for example, the information from early enrollees. But I think when you start thinking about the variants that appear in COVID-19, it really makes you pause to think about the equivalency of information from those people with the information coming from the concurrent controls. The other key element that I'm going to talk about today is this idea of trying to transition from a phase 2 component of the study to a phase 3. And what's shown here is a couple of agents, A and C, where they failed to graduate in some sense to phase 3. In other words, the signal for efficacy wasn't sufficient in the phase 2 component of the study. And then agent B is transitioning, hopefully or ideally, quite seamlessly into a phase 3 component. How seamless that can be actually is one of the challenges in this particular study. So, this, what we call the graduation decision at the end of phase 3, is based upon biomarkers or clinical markers that hopefully might predict the effect in the phase 3 setting on a hospitalization or death outcome. So this gets really at the surrogacy question as to how reliable these markers are for predicting effects in phase 3. Clearly a very difficult issue in the context of an emergent infectious disease. The other component of this phase 2 aspect of the trial is the intent to use the phase 2 population as part of the phase 3 evaluation. So although I'm going to refer to a transition from phase 2 to phase 3, it's important to bear in mind that the phase 2 study population is actually a component of the phase 3 evaluation. So just looking at the numbers here, we have 110 on a particular agent in phase 2. That's, and I'll talk about where that 110 comes from in a moment. And then the phase 3 is adding an additional 311. So giving a total of 421 in a phase 2 evaluation on the agent, and then being compared to a similar number in the control arm. And at the end of the study, there's a confirmatory evaluation based upon this combined population. So that's the evaluation, the effects of the agent on hospitalization and death. So as I mentioned, this platform study was really set up with the intent of evaluating multiple agents. And what's being done in this particular platform study is to evaluate each agent in its own right compared to a control group, in this case, a placebo control group. The study wasn't designed with the intent of comparing among the agents. So it's not adaptive in the sense of trying to home in on a best agent. And there's no response adaptive randomization in this trial. So there are some other platform trials for COVID-19 where there's more of an intent of trying to identify a best regimen. The intent here was to partner with lots of companies to test out each agent separately in an efficient manner. So not to identify or to compare among the agents. And that was thought to be very important in the context of trying to develop lots of options for treating COVID-19 and for facilitating partnerships with pharmaceutical companies and biotech companies. So when we talk about type 1 error control in the phase 3 setting, we're doing it separately for each agent versus the control. And there's no concept of trying to control the family-wise or experimental-wise type 1 error rate across lots of agents. So that's obviously a potential point of controversy, but the FDA was quite happy with this approach, and I believe they've incorporated it in their guidance for platform studies for COVID-19. So this is a very busy slide, but it tries to give you just a quick flavor of the initial design of the study. So the initial regimens that came into the study were either infused or injected. So they're typically a single infusion or a single injection, or maybe 2 infusions or 2 injections very close together, so an hour apart or something like that. The study has actually evolved, and it now includes regimens which are given through inhalation, so putting a mask on someone's mouth and nose to administer the dose. There's also oral dosing as well of traditional pills. So we have agents with different modes of administration. The core of the study is really the first 28 days of follow-up. And when the study was originally designed, it was really quite intensive. And this reflects the complete lack of knowledge that was out there about outcomes and interrelationships between outcomes for different, for COVID-19. So it had blood and saliva, as well as nasopharyngeal swabbing. And so these tests were focused on measuring how much virus was in different compartments of an individual's body. We also had the participants self-swab, not right high up in the nose, but lower in the nose. So these are called anterior nasal swabs on a daily basis, again, to measure virus. So these were being done at clinic visits, but we had daily measurements also to try and assess that as a potential biomarker. And then the participant also collected or recorded their symptoms on a daily basis during the first 4 weeks of follow-up. And then there's a long-term follow-up. Initially, it was out to 24 weeks. It's now been extended to 72 weeks. And this reflects in part the long half-life of some of the agents that are being used, so combinations of monoclonal antibodies in particular, but also a growing interest in understanding the effects of treatments on what people call long COVID. So the long-term symptoms that some patients experience with COVID-19. So phase 2 was quite intensive. Obviously, it also has an intensive safety focus. And then phase 3 is less intensive. So the visits were at baseline and at 4 weeks. We relied on individuals, the participants, to self-swab the lower nose to get some information about the virus. And then they also completed their study diary. So the outcomes of interest in phase 2 are more virology-based, looking at symptoms, also looking at oxygen saturation, so how much oxygen is circulating in someone's blood. And then in phase 3, really shifting the focus to the key clinical outcome of hospitalization and death. So this difference in intensity becomes important when we're trying to think about sort of efficient study designs, and I'll come back to that briefly shortly. So just to give you an idea of where the study is at, I'm actually not going to present data because there's very little data that's actually been presented from this study so far because we're in ongoing evaluation. But there was a recent press release a few weeks ago of one agent that did go right through phase 2 and phase 3. And in phase 3 showed a very substantial effect on hospitalization and death. So a 78% reduction in hospitalization and death in this outpatient study population. So a very successful outcome for that particular agent. So this is a combination monoclonal antibody agent. So I'm really going to focus on two topics which go back to those schematics. And the first one is trying to think about a control arm in a platform trial where you've got treatments that are administered in different ways. So I mentioned earlier, some treatments are infused, some are injected, some are taken nasally as a spray type of thing, or inhalation. Some are sort of more standard pills. So you can think about having multiple placebos in terms of trying to construct a placebo control group. So I'm going to talk a little bit about that. And there's actually a very complicated randomization system which facilitates this. So I'm going to illustrate some of the complexities of actually doing a platform trial in this setting. So just to remind you, for the comparison of any agent, we're comparing it to a concurrent control group of participants that were eligible for this particular agent. So it was thought, as is often the case, that having a placebo control was really, really important. And you know, some of this is based upon experience with influenza. Obviously a similar disease in some sense, but perhaps less, leading to less hospitalization and death. What we knew from influenza trials is that there was considerable value in having a placebo control, particularly in terms of things like retention, completeness of evaluations, getting these nasopharyngeal swabs. They're quite intensive, as some of you may know if you've been through that process, and trying to get them repeatedly over time is potentially difficult. And also there's an intensity of clinic visits, and obviously COVID is like influenza is you know, a very fatiguing disease, and getting people to come into the clinic is not always easy, particularly on a regular basis. And then key outcome is obviously measuring symptoms on a daily basis. So having complete symptom diaries is important. Also, it is really important. We often think of hospitalization as a somewhat objective outcome, but it really is quite a subjective outcome. It is a clinical decision, and you know, observation of people through a night in hospital is not an uncommon thing to do. So there was concern about the potential for subjective biases if a placebo wasn't incorporated into the study. So this raises you know, quite a difficult issue when you've got agents which are administered through different modalities. So the infusion, injections, pills, inhalation, etc. And also having different durations of administration. So when the goal was to rapidly try and evaluate as many agents as possible, the concept of having participants take placebos for all of the agents that are in the study was really not something that could be entertained. So it's very complex, therefore, to do a fully double-blinded study. So the solution was to use a partially blinded approach. And it's partially blinded in the sense that it does use a placebo, and participants don't know if they're receiving a placebo or an active agent but they're not having to take, you don't have some participants taking lots of placebos. And the way we achieved this was through quite a complex randomization system, which you'll see is somewhat complicated, but it can be successfully implemented. And we worked with PPD, and they did a great job at developing this system. So, this schematic tries to capture what's being done in this particular trial. So it actually involves two randomization steps. So a first step here, where participants are randomized to what we can think of as agent groups, so group A, group B, or group C. And then a second randomization, where the participants in group A are randomized to agent A or the placebo for that agent. Participants in group B are randomized to an agent B or a placebo, and in group C to agent C or placebo. So this is the blinded aspect of the study, but participants know whether they're being assigned to potentially get agent A or its placebo, or potentially get B or its placebo, or C or its placebo. So that's why I refer to it as a partially blinded study design. So the blinding is about an agent versus its own placebo, but they know, for example, whether they're getting an oral pill or not, versus an infused agent or an injected agent. So that's the basic two-step randomization. And then the ratio in this second randomization, sorry, I should say that the ratio in the first randomization is just 1 to 1 to 1 between the different groups. In the second randomization, in this case, it's 3 to 1. And the 3 is coming from the fact that they could have ended up in 3 different groups initially. So why do we have that ratio? Well, we're trying to create, in the ultimate analysis, a group of participants who are randomized to receive a particular agent, say agent A, and compare it to a group of participants that received any one of the, in this case, 3 possible agents. So the 3 to 1 ratio here gives a comparison where the 2 groups are roughly of equal size. So the contribution to analysis is we compare all participants that received agent A to the combined placebo groups. And so each of these placebo groups is smaller than the active agent group, but when you combine them together, you get 2 groups of similar size. So this is the actual comparison that contributes to the analysis. So we often refer to this as a combined placebo group or a shared placebo group. So the point here is to get similarly sized or similar numbers of individuals on an agent and also in its concurrent combined placebo control group. So this does introduce complexity in implementation. It obviously means that in the informed consent process, these participants need to be made aware that they can receive any of these particular agents or the placebos for those agents. But what we actually found, and I raised this question recently with on a clinical site call, it turns out that this is actually a potentially attractive design for participants as well, because they have more of a chance of getting a potentially active agent than getting a placebo. So there are, although the consent process is definitely more complex, there is that attractive element. The caveat, or another caveat, there's no free lunch with efficient design. So another caveat here is that the combined placebo group mixes placebos with different modes of administration. So, from an efficacy point of view, particularly in terms of thinking about virology, that wasn't thought to be an important issue. but obviously it could potentially complicate safety analyses, and potentially it could affect the symptoms that the participants are experiencing and recording in their diaries. So that is a definite caveat with the design. Okay, so there are some other complexities with this, and what I didn't mention on the previous slide, when I talk about participants, it's possible that participants might not be eligible for all of the agents. So it's important to appreciate that there is an assessment of eligibility. We try to standardize almost 100% eligibility criteria, but occasionally a specific agent might require additional specific eligibility requirements. So really, we're talking about randomizing participants that are eligible for all three of these agents. So built into the randomization system is also the possibility that some participants might be eligible only for, say, two agents, A and B in this example. And then the randomization takes account of that by only allowing these participants to be randomized to two groups. And then we have the same sort of ratio here of 2 to 1 because there's only two groups to agent or placebo, agent B or placebo for B. So in essence, there's a stratification in the randomization system which takes account of any differences in eligibility requirements among the agents. So again, it's another complexity. And when you have, in this schematic, I just have 3 agents. We had, I think, 7 simultaneously at one time. And you can imagine that there are potentially lots of permutations of this. So though in practice, that was a relatively small issue. The other thing, and this one nearly caught us out, is that, as I mentioned earlier, the set of evaluations in phase 2 and phase 3 is different. So when you think about in this schematic, if we have a participant, so this is time on the sort of horizontal axis. If you have a participant coming into this study around here, they could go to agent C or agent B, or the placebos for those agents. But the agent C in this schematic is in phase 2, agent B is in phase 3. So the sets of evaluations are different for those participants. So that's another complexity. So let me just illustrate that. So again, this is going back to the first schematic of the randomization. So we introduced the idea of having to think about eligibility to the particular agents. But we also need to think about the fact that this first randomization to different agent groups is also potentially assigning them to different sets of evaluations. And I see I've actually got a, so in this example, I'm assuming that all 3 agents are in phase 2 at the same time. So that makes it straightforward. But if we instead have 2 agents in this example, phase 3, and 1 agent in phase 2, then we need to recognize the fact that phase 3 wasn't doing the virology assessments in an intensive way. So participants that are randomized to the control arms for these 2 agents, A and B, won't be getting the virology measurements in phase 3, and so cannot contribute as part of the control group to the phase 2 evaluation of agent C. So, so this is a further complication that needs to be taken into account. So the informed consent process needs to describe not only the different agents, but also the potential different schedules of evaluations that participants might encounter, depending on whether an agent is in phase 2 or phase 3. So I just put a couple of comments at the bottom here. In our trial, participants couldn't select among the agents they wanted to be randomized to. So it's sort of a package deal. If you're coming into the study, you need to be willing to consent to all of the agents that you are eligible to receive. It would be possible in practice to allow participants to have a choice, and that could be built into the randomization system. It's really sort of an extension of restrictions on eligibility, but it wasn't something that we did in this particular study. And similarly, clinical sites doing the study couldn't pick and choose which agents they wanted to evaluate, with the exception that for one or two products, there were practical issues such as sufficient availability of the regimen to be made available at all of the sites that were participating in the study. So what I've tried to show you is that we often think about platform trials as being very efficient mechanisms for evaluating regimens, but they are complex. There's no doubt about that. And the idea of having a placebo control group in this study was definitely a complex issue in terms of the randomization system and knock-on effects to the consent process and so on. So I just put a couple of bullets up here as a sort of critique of this. So the first three agents in ACTIV-2 actually came in sequentially, so there wasn't that complex randomization system. But the next 7 agents had really significant overlapping enrollment in phase 2. And this is where we really see the benefit in terms of that shared or combined placebo group with roughly a 25% reduction in total sample size that was needed to evaluate these agents. So the caveat of that is, for a given agent, about half of the participants in that combined placebo group were on the agent's own placebo, and hence had the same mode of administration, so an infusion, for example. But the other half were on the placebos for other agents, so potentially different modes of administration. So I mentioned earlier some of the problems that might be accounted in sort of safety analyses, maybe analyses of symptoms, by having different placebos with different modes of administration. The fallback position is obviously to restrict the analysis to the subset that were on an agent or its own placebo. And this is still a reasonably powered comparison. It's a sort of 2-to-1 ratio of number of participants in active arm versus the placebo arm. So there's definitely some loss of power if you have to fall back to that, but it's not as bad as it might first seem. What's unclear in thinking about the complexity here of trying to compare lots of agents against a shared placebo, it's unclear how the complexity of the consent process versus the sort of favorable aspect for a participant of an increased chance of getting a potentially active agent rather than placebo might have affected the rate of enrollment. We don't really have a good handle on that. So we can't really assess well whether it would have been faster to have done separate trials, separate standardized trials for each of the agents that came into the study versus having this shared placebo concept. And I should also mention, I'm not going to go into details here, there are some practical issues in protecting the blind when you have a shared placebo group. So it's just something people have questions about that they can always ask me in the future, but there are some practical issues there. So the other thing I'm going to talk about in the remainder of my time is this idea of a potentially seamless transition from phase 2 to phase 3. And it's really about how to decide and when to decide whether an agent graduates. And my comments really here are obviously have to be taken in the context of that we were designing a trial for a disease that we knew next to nothing about. The early information was very much focused on hospitalized patients, and there was extremely little information that was available about the broader population of people that had COVID-19 but weren't hospitalized, and that was the target population for our study. So key outcomes in phase 2. As I mentioned earlier, viral shedding is one. We measured that primarily in nasopharyngeal swabs, and that's eventually what we simplified the study to focus on. So what this is doing is it's measuring the level of virus by a PCR technique. And obviously these assays were being newly developed as the disease or the pandemic was sort of rolling out. So we didn't even know much about the performance of the assay that we were going to use. We do know now that the particular assay we have is extremely good. So the concept here, and this gets at sort of surrogacy type issues, the thinking was that lower levels of the virus would predict lower risk of hospitalization or death. That seems fairly obvious, perhaps, but it's important to bear in mind that we're measuring the virus sort of in the upper nose, and maybe it's the virus in the lungs that really matters. Very difficult to measure that there, particularly in the outpatient setting. So there's no guarantee that what you're measuring in the nose might be a good predictor of risk of hospitalization and death. And the other important aspect of this, obviously, was that the thinking was that the agents that we were looking at would have an effect through antiviral activity, in essence, by lowering the levels of the virus. Again, with the caveat that we're measuring it in one compartment, and there are other compartments that may be more important. And then also in phase 2, as a sort of key clinical outcome, and this was based upon what was done in influenza trials, looking at daily symptoms recorded by a participant in a diary each day. The thinking obviously here was that people that might have shorter duration of symptoms might be at lower risk of hospitalization or death. As I mentioned, there really is, or was, extremely limited data about these types of measures in the population we were focused on. And I have to say, even a year later, or a year or so later, there's still a huge uncertainty about the interrelationships between these markers and hospitalization death. And it's kind of notable that the FDA still hasn't made a move on sort of saying much about the viral markers or the symptom outcomes as potential surrogates for hospitalization or death. So it reflects still a lot of uncertainty here. So I just put this up just to give you an idea of what we had available to us when we were designing the trial. And this is results from a single trial that was published in the Lancet in May 2020. And what it's showing is on the y-axis, the log viral load in nasopharyngeal swabs for a treatment group versus a control group plotted against time in days. And this was data from hospitalized patients, so a very different population from what we were dealing with. But it did give us some idea of how quickly viral loads declined over time, and the type of difference between a control and a treatment that might occur. So this difference is on a log base 10 scale over 1. What this graphic doesn't show is the fact that roughly about half of participants actually had undetectable virus in this treatment arm at 7 days, so 1 week. So there's a lot of imputation going on in the background behind this type of plot, and that's an important statistical issue in these types of studies. So it's a busy slide, but just to give you some idea, my schematics earlier had sample sizes for phase 2 of 110 participants on an agent, and then the control group would be a similar size. And you know, in the almost complete absence of knowledge about what might occur in this particular trial, we took a very simplistic approach to picking that sample size. It was partly driven by the need to get a reasonable safety database, but it was partly driven by the idea of just getting a reasonable level of power to detect a 20% absolute difference at any one of the measurement times in the proportion of the study population that had viral levels below the limit of quantification of the assay, so essentially undetectable virus levels. So it was a sort of a shot in the dark, if you like, but it did give some basis for where that number came from. So graduation criteria. So this is about assessing efficacy seen on markers in phase 2 and trying to decide whether to graduate or take an agent forward into phase 3. And what our approach, again, in complete absence of any knowledge about interrelationship between these markers and hospitalization or death, it was a sort of stab in the dark. But it was based on thinking about the probability, so it was a Bayesian-based analysis, so we're looking at posterior probabilities here. The outcome for an agent is better than the control, the placebo, by an amount of at least X, and X depends upon the marker you're looking at. So we wanted that probability statement to be greater than 0.6. So in other words, there's sort of some evidence that the agent has an effect greater than X, not overwhelming evidence, but some evidence the effect size is greater than X for a particular marker. And the markers we looked at were 3 virologic ones, and this reflects the fact that we thought these agents would have their effect through an antiviral effect. There are different ways of looking at the virology. So one was just looking at the absolute proportion of participants that had viral shedding below the limit of quantification of the assay at any one of these time points, 5 different time points. So that's a sort of yes, no.
Michael, sorry.
Yep.
Sorry to interrupt. There is one question that is related to that. So is the phase 2 primary endpoint on any of the listed, or is it possible that viral shedding can become less than the lower limit of quantification, but then increase again?
Yep, so it was done marginally at a given time point. So there are actually a huge number of assessments going on here. So if I just take this particular outcome, so it's just think of it as undetectable or not. We're looking or comparing the proportion undetectable on an agent versus placebo on each of these 5 different days. And you know, in theory, any one of those, you know, seeing a sufficient difference on any one of those days would graduate an agent. So there's a huge multiplicity issue here. There obviously would be a lot of correlation in these outcomes that we're not taking account of, but we had really no idea beyond that one trial of hospitalized patients, which is a very different population, when we might see you know, a reasonable proportion becoming undetectable, and further, when the differences between an active agent and a control might occur. So in essence, we're covering our bases, and we're looking for any evidence of antiviral activity. So there's definitely a multiplicity issue here. So this is looking at the virology dichotomized. We can look at it quantitatively, and that's fine. But the caveat here is that at some point, the majority of people will be below the limit of quantification of the assay, so it's harder to use the quantitative information there. And then we could also look at some sort of AUC, area under the curve, of viral levels across the 28-day period. So this is sort of looking at a totality of viral burden over that 4-week period. So just to reiterate, in essence, we're looking for any evidence of an antiviral effect of a sufficient magnitude. And these, what I've shown in red here are these X's, they're sort of targets. And the underpinning for these two was really that one single trial, which showed that a 1 log difference was plausible. So getting a half log difference may be evidence of an effect. Again, we have no idea, or had no idea, about how that might link with hospitalization and death. And then we also looked at symptoms in terms of duration of symptoms. Again, we had no idea how long the symptoms might last in participants. So these are sort of the core of what we had, but we also had a safety net. And I'll talk a little bit about this safety net because it's quite important in terms of looking for a 1/3 reduction in the proportion that are hospitalized or dying. So I mentioned huge uncertainty about these markers, or even whether they are markers of hospitalization or death. So we were looking for a signal in lots of different ways of what we thought was a reasonably sizable effect. And in essence, what this approach needed to show is that the observed effect needed to be a little bit larger than the target on any one of those markers in order to graduate. And that concept of you essentially being based on the observed effect is really quite conceptually appealing when you're discussing results with particularly with non-statisticians, but with clinicians. So there was a really extensive simulation study that was done, partly by one of the participating companies and then also by our group at Harvard, that showed that there can be multiplicity concerns, but because those thresholds were the X's, the targets were of a reasonable size, the probability of graduating an agent that was truly ineffective on all of the virology and symptom duration outcomes was actually really quite low, so probably of the order of 1 or 2%. And it's driven by the outcomes which have low precision. So the less precise outcomes drive that error rate. So let me just talk about the safety net, because this is an important concept when you don't know much. So the phase 2 trial, we were talking about 110 participants receiving an active agent, similar number in the control group. The phase 3 trial, which was designed to detect a halving in the hospitalization death rate, had a total sample size of just over 400 on the agent and 400 on the active. So there was, about a quarter of the information on hospitalization or death was actually going to come from the phase 2 participants. Remember that those were going to be part of the phase 3 analysis. So we wanted to avoid the situation where we were seeing no effect on the virology or the symptoms, or very minor effects, but we actually were seeing some evidence of an effect on hospitalization or death. It'd be very embarrassing to kill off a product and not graduate it in that sort of situation. So we're sort of covering our bases. So we did have this safety net that allowed an agent to graduate, having shown no or minimal effect on virology and symptoms, but was the observed effect on hospitalization death was at least a 1/3 reduction. Caveat here, obviously, is smallish numbers, an important safety net. So the problem with that is that it's an imprecise outcome. And imprecise outcomes, when you use those sort of Bayesian probability statements, where the probability, the posterior probability, just has to be bigger than 0.6. Imprecise outcomes make it easier, are the ones that are more readily met in that sort of situation. So we could show through simulations that there was a reasonable risk that an agent which was completely, in truth, ineffective with respect to virology, symptom duration, hospitalization, or death, had, in excess of a 0.15 chance of graduating, and it was driven by this safety net. So this was addressed really in the phase 3 setting by having quite an aggressive stopping guideline for futility. For those of people that are familiar with sort of stopping boundaries, here's our boundary that we were using in phase 3 for Z statistics, standard sort of O'Brien and Fleming type boundary for efficacy. But we had a futility boundary, which is really quite aggressive. So the intent here was to overcome this problem of potentially graduating an agent based upon a sizable chance difference, and then finding out that it wasn't particularly, it didn't really hold up in phase 3. So this futility boundary is relatively aggressive. It also protects for the situation where you're graduating agents based upon virology and symptoms, and they're actually poor surrogates for hospitalization or death. So the futility assessment in phase 3 was quite important. I'm going to skip that. So making graduation seamless is quite challenging when it takes several weeks to get the virology testing done and the results back and analyzed. So one approach that we adopted in the study was to do the phase 2 and then do a safety assessment that allowed the enrollment to continue sort of at risk, pending getting the virology and the symptom data together. So that was the concept of sort of making a somewhat seamless transition from phase 2 to phase 3. Obviously, the problem here is that you might then do your graduation analysis and find that the agent doesn't meet criteria. And in that sense, this is then wasted enrollment. So the rationale for that, doing that at-risk approach is lower if you have other agents enrolling in the study at the same time. If you only have one agent, then it seems more reasonable to pursue it. The other aspect of the trial that was particularly complicated here was the phase 2 study population is part of phase 3, and it's close to about a quarter of the total phase 3 enrollment. So we had the scenario where we might be doing a phase 3 efficacy interim analysis at the same time as the phase 2 graduation analysis. So how do you handle a conflict between those two analyses? So there were very clear criteria or stipulations in the protocol that the phase 3 stopping guidelines for efficacy and futility would take precedence over any graduation decision. So really focusing the assessment on hospitalization death outcome rather than the virology and symptom marker outcomes. So let me just say that we often think about platform trials as being adaptive. They're adaptive in this trial in the sort of phase 2 to phase 3 context. But probably the biggest adaptation that occurred during the study was the accumulation of information about when hospitalizations are occurring and what was happening with the virology. And these graduation criteria have actually evolved, and they're now much more focused on what's happening during the first 7 days of follow-up, which is when most of the participants have become undetectable with respect to a virologic outcome. And are also, that's when nearly all of the hospitalizations are occurring. So we now have implemented a different approach, which is much more focused on the day 7 virologic and symptom outcomes, and involves an interim analysis actually earlier during phase 2, focused on those data, with the idea of using that to decide whether to proceed immediately into phase 3 enrollment or not. So this is a slightly different approach for the transition, or the seamless, potentially seamless transition from phase 2 to phase 3. So I'll close just with this slide. And this is an ongoing platform trial. We now have effective agents in the US for treating outpatients. There is a limitation on how available they are, but I showed you the results from our own trial, but there are a couple of other trials which have identified effective agents. So the idea of continuing a placebo is no longer acceptable in a higher risk population. So what's happened in this trial is phase 2 has now shifted to looking at virology and symptom outcomes in lower risk people, so people that aren't eligible under the emergency use authorizations for those agents. And the phase 3, however, is still continuing as a separate trial, platform trial in a higher risk population. And that's now going to be active controlled and comes with all sorts of issues around doing an active controlled non-inferiority trial. So I'll stop there. I hope it's given you some idea of how you think about doing a platform trial with an emergent infectious disease. And hopefully it's given you a feeling for the complexities that these trials involve, which needs to be balanced against the sort of efficiency gains in terms of reduced sample size and having a single protocol to evaluate lots of different agents. So thank you very much.
Thank you, Michael. Thank you so much for joining us today. I know we're a little bit over time, but if anyone has any final questions, feel free to put them in the chat, and maybe we can address them in the next 5 minutes or so. Great. Well, if there are no questions, I want to thank Dr. Hughes again for agreeing to present. And I wish you all a happy Friday. So thank you all for attending.